Preeti Pancholi, if I said that right, talking about MALDI-TOF in clinical microbiological analysis. So welcome.
So thank you very much, Tim, for the invitation to come here and talk today, and good afternoon, everybody. This is my first time at the Pittcon conference, so I'm kind of excited to be in the midst of all you chemists that know a lot more about mass spec and everything else. And here comes a microbiologist who's going to be talking about all the wonderful things that are happening in my lab with mass spec at the moment. It's a very exciting time for clinical microbiology, for sure. So what I'd like to do today is just kind of touch upon three objectives, and I'll go into details of these, just to talk about MALDI-TOF in clinical microbiology and how we implemented MALDI-TOF for microbial identification and how we can't imagine life without it today, even though our implementation has only been about a year old. And then the impact that this is having on our patient care and things that continue to excite us to do even more with MALDI-TOF. So just a brief introduction of my place of work. I work at the Ohio State University Medical Center. It is a quaternary care medical center that really deals with a lot of different types of patients. So we have three major hospitals as a part of this hospital setup. So we have the Ohio State University Medical Center, Maine, that also has a Ross Hart Hospital. We have the James Cancer Center, and then we have the OSU East hospitals. And so we are overall about a 1,400-bed hospital facility, and the Ohio State University Medical Center is 980-bed, catering for general, medical, and surgical facility. University Hospital East also is general and surgical facilities and has wound care specialists as well. James Cancer, as you can understand, is really all geared towards cancer patients. And then we also service about 37 outpatient clinics. The reason I show you this is just the whole intensity of, I guess, just the workflow and the patient population that comes into our system, and that from these different hospitals and the clinics, the clinical microbiology all sort of filters into our lab in clinical micro. So right at the foremost, you should know that microbiology is a culture-based science, okay? So we still rely a lot on cultures. I can't imagine not ever having cultures, because if you look at a patient specimen that comes to us, we have to kind of find out from this culture what is important for us to tell our clinicians. We don't want to be giving out normal flora results. We really need to focus on pathogens, like looking for needle in a haystack, if you will, to really give our relevant answers and not irrelevant answers. So we really do have to end up sort of trying to culture a lot of these different sample types, because not all these sample types are sterile, okay? From sterile sources, yes, you can certainly make certain more predictions. But with non-sterile sources like your BALs, your respiratory, your sputums and all of that, you really have to find out the relevant pathogen. So culture still remains very integral for clinical microbiology. Even though we've implemented, rapid technologies that are based on RNA and DNA, nucleic acid technologies, sequencing and all of that, and we do a lot of mix and match of different technologies in the microbiology lab to give answers. However, we still have to rely on culture to a great deal. So we rely on culture conditions, how quickly does the organism grow, what special media do we need to have the organism grow on, what is the staining characteristic by gram stain, what's the microscopic morphology of the organism, and then we also rely a lot on biochemical phenotype of the organism in which to get an answer. And when you start now talking about biochemical tests for organism identification, as you can imagine, this is not an exact science, right? You're looking for a change in color or a change in something with your matrix here. So it's a very inexact science. It can be time-consuming, and sometimes you get a probability of 55%. What does that mean? That really doesn't give you very much of a clue as to what the organism is. So the strategies that have sort of now really playing a major role in microbiology are, how do we get rapid identification to occur for pathogenic organisms so that we can give these answers out quickly to our clinicians, and that which can have rapid impact on patient care and can also decrease costs in the lab. So time, cost, and patient care have become really relevant in micro. And to do that, we've actually been utilizing three different technologies. One is the MALDI-TOF, which I'm going to talk about in detail today. We've also implemented a number of nucleic acid-based tests, and then, of course, sequencing, which we have in place as well. So with regards to the MALDI publications, as alluded to over a previous speaker as well, that there really is a real explosion of papers that are coming out and relevant to clinical microbiology in this particular arena. There's more than 250 peer-reviewed publications. They may not sound like a lot, but believe me, as compared to the previous years, this is an area which is rapidly escalating. And the groups of bacteria that are relevant are your gram-negative rods, your yeast, your anaerobes, your acid-fast bacteria, your molds, your fastidious organisms that take forever to grow or just never grow out on culture media, like your nutritionally variant streps. By the way, I've spelled these acronyms out down here in case you're wondering what are these acronyms. Bioterrorism organisms, your HACEC group of organisms that cause smoldering infections, and these are 5 different groups of organisms listed here. Which is your Haemophilus, Cardiobacterium, Eikenella like organisms. And then, of course, you have your bioterrorism like Brucella, Francisella, and you even have your parasitic organisms now that can be detected by MALDI-TOF. So the other papers that have been coming out have been on lab implementation as to how to implement these type of methodologies. Because remember, we are a culture-based science. We know how to work with RNA, DNA. Now, all of a sudden, we're in this realm of mass spectrometry. We're dealing with chemicals, and we're dealing with all these different, aspects of safety in the lab, which to you probably comes inherent since you're clinical chemists. But for us, we really do have to sort of pay attention to some of these differences that are now coming our way with mass spec. So there are papers out there now for blood culture direct analysis, where if you have a blood broth that has been incubated, is now growing an organism, can we take that broth directly and identify the bacteria? So we don't want to grow it on culture media if possible. Right from the blood broth, can we identify the organism? Likewise, can we directly identify organisms directly from specimens like urine cultures, which is a large part of sample types that we get, in the lab. Then there are other subtyping of the organisms. There's development of resistance protocols, because along with identification, you also want to be able to tell your clinician what to treat with, what antibiotics to use. And so really these things need to go hand in hand with your organism ID. And then virulence factors is another very interesting area of research as well. So MALDI-TOF mass spec, right up front, I'll tell you the bright points with MALDI-TOF. Rapid turnaround time, okay? We're talking minutes here, and I'm sure you all are used to it. I was kind of a little amused when I was listening to a couple of speakers that said, oh my God, 10 minutes in the lab is too long an assay. I mean, you're really not, that's not a good thing. We need to cut it down to less than 10 minutes. Well, I can tell you we're absolutely thrilled to be able to get an answer in 6 minutes in the lab, because previously we've just waited literally days, if not weeks sometimes, for the slow-growing pathogens. So for us to be able to get our answers, that same day within a couple of hours, especially if you have to do certain extractions, is pretty phenomenal, okay? Because we just never had this ability to do this before. So it's very economical, and I'll go into some of the economics of this, except for the instrument that's very expensive, but the actual reagents are really very, very economical, and the instrument more than pays for itself. And it's automated, high throughput, minimal consumables. So you can see there are just some really, really important points listed here that make it very attractive. And the beauty of all of this is that for microbiology analysis, you just need a single colony of an organism. So even if you have a plate that's growing a mixed population of bacteria, so long as you have one nice-looking, good-looking, isolated organism, on your plate, you can just touch that organism and get your, profile right off of that. So you don't have to wait to subculture and all of that stuff that also adds to time and delay. And then the other thing, as I mentioned, that we in microbiology, we have a lot of different types of culture media that we use to encourage the growth of the organism. And the beauty of MALDI-TOF is it does not care what media you grow your organism on, okay? So you can literally utilize a lot of different medias in which to actually test MALDI-TOF. The only criteria here is, since we're looking at ribosomal proteins, that you have an organism that is relatively fresh, which means less than 24 hours old, in which to do your MALDI-TOF off of, because you're looking at ribosomal proteins. So we have 2 different mass spec instruments that are currently commercially available. One is the Shimatsu or the Vitek mass spec, and the other one is the Bruker Biotyper that are commercially available. Now, this is a tabletop model. The Bruker and the Vitek is a floor model. And both have recently obtained FDA approval for certain organisms that are present on their databases, okay? So it is wonderful news that there are these systems now that they have FDA approval for some gram-negative, gram-positive bacteria and yeast for the Shimatsu system and gram-negative bacteria for the biotyper system. Now remember, they only have clearance for certain handful of organisms, and there are a large diversity of organisms out there. But this is very encouraging news, especially for people who've always been afraid of mass spec and have always been afraid of getting a system in the lab that is more research use only. We started off with MALDI 2 years ago when it was labeled research use only, and once it came to the lab, we knew it was never leaving us ever. But it's just gratifying to see that the progress that's being made now in terms of FDA approval. So simplistically, how does it work? Well, it's really a very simplistic workflow if you think about all the complicated things we've been doing in the microlab in the past. So you have your organism that's growing on your primary plate media. You take your one organism, one colony of that organism, select the organism, and then you smear it onto this MALDI plate. Okay, we use the 96 well grid plate, that's stainless steel. You can also use disposable 48 formats and so on. You smear your organism in a thin layer, and then you add the MALDI matrix, put it into your MALDI machine, which will then, there'll be a laser inside, and it'll generate a MALDI-TOF profile spectrum. The spectrum is then interpreted with the databases that also come with the MALDI instruments. So each of these instruments that I showed you has a database that comes along with the instrument when you purchase it. And then you identify your species based on a score match that you get. So you can imagine now, your one sample in 5 to 8 minutes, you're getting your answer versus having to wait, for a long time with biochemical methods. So the setup is really simple. Just look at this. All we've got here is Petri dishes, and we've got your MALDI plate here, the steel grid plate. You've got your high-tech equipment, toothpicks, in which to touch your organism. And you've got a few little chemicals like your formic acid and your matrix and so on. And you have a centrifuge, in case you need to spin down and concentrate your organism and so on, and ethanol. So very, very simplistic setup. Here is a technologist. She's touching with a toothpick one isolated organism from a plate that she wants to do MALDI on. And so as you can imagine, target preparation is something you need to pay a little bit more attention to. It's not simply that each and every organism will work well with a direct smear method. I would say a large number of them do, and then there are certain groups of bacteria that require a little bit more than a direct smear method. So with a direct smear method, you touch the organism and you kind of smear it lightly onto your matrix plate, onto your MALDI plate. And we generally tend to make 2 different concentrations, one slightly heavier than the other, so we make sure that at least one of the spots works in case one is too low. And then for organisms that are encapsulated or that may be mucoid in nature, it's really helpful if your initial spot doesn't work that you go ahead and you do a chemical extraction method, which is simply resuspend the organism in ethanol, spin it down, and then you add your formic acid and acetonitrile. And then you are ready to look at your supernatant, and then you smear that supernatant onto your MALDI plate. And the advantage of doing the chemical extraction method has been that we've gotten some really nice looking clean spectra as a result of just spending an additional few minutes by getting this little chemical extraction method going. And also because of the fact you're adding ethanol, you're also helping kill the organism. So it's less of a biosafety issue if you're working with it outside. So since not all microorganisms are created equal, we also work on mycobacteria a great deal in the lab. So we have a semi-P3 lab that works on mycobacteria, because this is a special group of organisms. And you all may have all heard of Mycobacterium tuberculosis, that is the causative agent of TB, tuberculosis. So we really have to handle these organisms very carefully in a semi-P3 lab. And now that we, when we first had our first MALDI, obviously we cannot have a MALDI in every areas of our lab, in the anaerobe area, in the aerobic bacteriology, in the mycobacteria fungal lab. We only had one MALDI at the time. And so we wanted to make sure that we were going to really process mycobacteria very carefully, which would mean that we, end up, first adding ethanol and then heat killing the organism at 95 degrees. and then again eluting with ethanol, and then adding your zirconium beads, because they have very thick waxy cell walls, okay, with mycolic acids in it for mycobacteria. So you really want to make sure you crack open that cell wall really well, and that you are then able to get the proteins out, which then you can add your formic acid and so on, the same steps as I showed you previously, and then spot your supernatant after going through the process onto your target plate. So you can see it takes about less than 1 hour once you have a culture to identify in which to get your MALDI answer. So previously, when we had a mycobacterial organism to grow, it would take us, when we used to do biochemicals, it used to take us weeks. And then when we switched to sequencing, it used to take us days. And now, because we don't have a sequencer in-house, but now that we have a MALDI in-house, it literally takes us that same, like a few hours from the time we have an organism growing to the time we can get the organism identified. Pretty amazing, okay? So, and then when we think about doing direct blood culture analysis, this is another application that is being looked at by several labs. And so, again, this would mean that if you're taking a direct blood broth that has organisms growing, you want to remove your blood cells, serum proteins, and hemoglobin so that you can really get at the bacteria. So there are simplistic methods in which to lyse the red blood cells and to really purify your organism out. And so Bruker sells a kit called Sepsi-Tiber kit, which you can buy, or you can make your own homebrew version of reagents in which to get to the bacteria. And this takes about maybe 5 to 10 minutes, but it actually gives much better spectra. So what you do is, so now you've got your organism onto this target plate. You add your matrix, which is your HCCA seems to work really very well for a lot of our microbiology analysis. And then you're now ready to do the laser part, which is now you're putting it into the instrument, and that's the laser that's shining on your spot. And then here's your desorption that's occurring as a result of the energy that is transferred. And then now here's your time of flight occurring. This is kind of cool. Bruker has kindly loaned these slides to us just to show you the mass, the movement of the different protons that are happening here. The smaller particles moving faster than the larger particles, and a separation occurring on mass and charge ratio here. And then what you end up getting in the end is this very nice spectra. That is generated from the ribosomal proteins of the organism, and the spectra is then, looked at for the database to find a good match. And so my talk today is going to be more with the Bruker system, because that's the system that we have adopted for the last couple of years in our lab. And with the Bruker system, you get a very nice score matching. If you have a nice match with your spectra, you get a green label, which means if you have a score of 2 to 3, you can call out a secure genus and species identification. If you have a yellow score, it's kind of like caution. It is a probable genus identification, so you don't get the species part of this particular organism. And then if it's red, it says, wait a minute, there's unreliable identification here. So either your spot didn't work, or the organism needs extraction, or there's nothing in the database that matches that particular organism. Okay, so it's really kind of cool. And what we've tried to do is we've tried to stick with this score of 2 to 3, which to us designates a very good score. There are labs that are working on certain organisms, and they may choose to go down from the score and maybe accept 1.7 as a cutoff for giving you a secure genus and species identification. Like I said, this is an open area of research, okay? So if your lab can feel confident that a score of 1.7 is good enough and can generate a good species, genus and species, well, you just really need to prove that in your lab. We've tried to adopt extraction protocols, and we feel better if we had a score match of 2 or higher in our lab. So the databases are extremely relevant and extremely important, as you can imagine, for any of your MALDI-TOF applications. So the biotyper actually comes with a reference library. The reference library contains spectra for bacteria, mycobacteria, yeasts, and molds. They also have a security-related library, which is relevant for bioterrorism organisms, which is also something I think one should have in the midst, because if you think about bioterrorism events can happen, even organisms like your Francisella and Brucella are listed under BT, bioterrorism organisms. But these are organisms that you can encounter in the lab with just, some sort of a history that's on a patient. So you really don't want to be exposing your lab technicians to any of these organisms if you have a way in which to identify them quickly so that there's less processing and less of biochemical tests being set up for these organisms. So it does make sense to get both of these databases which come along with your instrument. And for the Bruker, they have about 5,600 database entries that includes about 2,000 different species. And again, when you think in terms of this different species, only about 100 gram negatives or so are FDA approved at this time. So all the rest of these other organisms, really, it will depend on the lab's ability to feel confident that the result that they're generating as a match for these other organisms have been tested and tried in the lab to some extent, right? Because those other organisms are research use only. They're not FDA approved. Except for these small handful of organisms. So there is work to be done by any clinical microlab that really wants to utilize this for a broad spectrum of organisms. And the other interesting thing you should know about is that it also has an open library system, which is a library that you can develop on your own if you have organisms that are not represented in the database. So we've used this open library a great deal because we used to do sequencing a lot previously because as you can imagine, we encounter a large number of organisms that, quote unquote, have not read the book, or they would not pan out with any system, either by biochemical method or by nucleic acid testing. And so we used to get these sequenced regularly with different primers and so on. And then once we had this repertoire of organisms, we systematically did sequencing and we did the spectra on these organisms and we stored them for our own databases. So it is really, really nice because when you're querying these organisms on the database, you can query all of these different libraries all at the same time, okay? So it tells you if it found a good match or not, and so on. Now, another great resource is the NIH database, which is available upon request from the NIH, because the NIH has been working on the Bruker MALDI system for a few years, and they have developed their own databases for how to identify organisms, especially the molds, for example, and mycobacteria, nocardia, okay? So it's really kind of cool to have that ability to ask NIH to give you access to those libraries, and then that way you can query their library as well along with your own library. Okay, so it's taxpayers' money being put to really good use here. So how well do these systems work? Okay, we have the Bruker biotyper, we have the Vitek Shimatsu mass spec, and labs have undertaken studies with these systems. And with both of them, when the same organism was tested, you can see that they agree pretty well for a large number of organisms. So upwards of 92 to 93% of the organisms were correctly identified by both of the systems. Now again, they're testing a certain handful of organisms here. There were some organisms that were not identified on either of these systems, and some of these include some of your anaerobes that don't do very well, some of your streptococcus species. For example, strep pneumonia is not identified very well with Bruker system or with Vitek. And then, and also your E. coli, which is an enteric organism, cannot be very well distinguished from Shigella. So now you know Shigella is something you don't want to be mixing up with E. coli. You want to be able to distinguish both of these organisms. But because they are so taxonomically so closely related that you cannot separate them based on their ribosomal proteins, so they tend to give you the same answer, okay? So in that situation, the lab needs to know the limitations of each of the systems, what it can identify and what it cannot identify with high confidence, and be able to set up ancillary tests in which to be able to dissect out these organisms. So it's really not difficult to do that because, as you know, we are used to setting up biochemicals in the lab and certain ancillary tests. It's just that don't put everything away, that you're never going to utilize some of these other reagents till you are sure that your organisms are panning out well. And then, of course, sometimes in the databases, some organisms may be misidentified, so that's just something else to keep a very open mind, that don't believe everything you see. Make sure that when you have a match with your organism in the database, you're looking at the organism, looking at the match, and you're looking at some ancillary tests before you really believe what you see. So there was a very nice study that was done by Johns Hopkins Lab, Tan et al., and they showed that, okay, what is the impact of MALDI in terms of time to identification? And so these are your organisms listed here, a number of different gram-positive, gram-negative organisms that generally would take you at least 3 to 4 days, if not more, in which to identify. Now can be identified, much sooner. So your red bars here represent MALDI-TOF based identification, and your blue bars are other tests that are routinely done standard protocol-wise. So you can see things that took 3 to 4 days can now be identified at least 1.5 days sooner than as using your standard protocol. So this is a really big advantage. And the other advantage is for also slow-growing organisms like your mycobacteria and your nocardia, that time savings is even more significant than what you see for some of your standard aerobic and anaerobic organisms. And in terms of the cost, to see how cost-effective is MALDI-TOF, here you have your annual reagent costs for your standard protocols that were being used in the lab, and also now compared to your MALDI-TOF protocol. And you can see your annual reagent costs, your annual labor costs, is much lower for MALDI-TOF procedures. You do have some fixed annual costs, which is your service charges that you have to pay on an annual basis are pretty high, along with your quality controls and other tests that you have to do in which to identify the organisms. So the fixed costs would really refer to some of those extra costs that you have with MALDI-TOF. But overall, nonetheless, it is about a 56.9% drop in labor costs over a period of 12 months if one were to introduce MALDI-TOF. So a MALDI-TOF instrument currently costs around $200,000, and I think a good point to make to your hospital administrators is that within a couple of years, you definitely will be able to recoup the cost, if not sooner, depending on the kind of volumes that you test in your lab. So in terms of implementation of MALDI-TOF, like I said, a lot of labs are considering bringing this on, and some and several have already implemented MALDI-TOF. For us, it was really important to look at how well will this be implemented in our setting. Is it a good fit for us? Because we are a large hospital with high volumes of culture, so we said, yes, this is absolutely really going to be worth our while to work on this. When we got it, it was completely a research use instrument, which meant that we had to do a very rigorous in-house verification against standard lab identification systems. So we use a number of different systems like the microscan, the different biochemicals, probes, sequencing. We wanted to make sure that the organism identification that would be getting out of the MALDI would be accurate. So we put to test a number of bacteria, mycobacteria, yeast. We tried several different extraction protocols, and also looked at what this would mean for our workflow and procedures that we had in place, because this was going to change our workflow quite a bit. So this is these are the organism repertoires that we looked at: gram-negative, gram-positives, and so on. You can see a large number of organisms, also yeast and mycobacteria. And overall, our genus and species level identification were quite high. So genus level 96 to 99%, species level about 85 to 97%. So the considerations in front of us were, is this going to be considered as something that we need to bring on house and will be having some cost savings in patient care? And how do we convince our hospital administration to come up with this money so that we can actually go ahead and buy this instrument? So what was really advantageous for us was we have an antimicrobial stewardship program in our hospital. We meet once a week. And we approached them, and I'll show you the different members that are part of the antimicrobial stewardship team. And they were just super excited about the idea of being able to get results quickly from the microbiology lab. And then we asked them the question, if we give you rapid results, are you going to be able to utilize them rapidly as well? Because what use is a rapid micro result if it's not going to be utilized, in rapid real time for clinical use? So we really wanted to ask that question of them as well. And so our antimicrobial stewardship team comprises of a group of individuals that includes an ID physicians, ID pharmacists, clinical microbiology, infection control, the LIS folks, the epidemiology folks, and of course, centered is the patient. This is all regarding patient care, that all of us are here as part of team members. It's a stewardship team. We meet once a week, and we go over all our issues and problems with drug resistance and getting answers and trying to see, how do we optimize, doing the things that are going to benefit patient care. So what we did when we started to work on MALDI-TOF, we had a plan where we would be sending our ASP pharmacists 2 daily reports of all positive respiratory and blood cultures. And the pharmacist would then review the patient's medical charts to ensure appropriate initiation or de-escalation of antibiotics. So that would be their job. If they got these 2 reports from us, once in the morning, once in the afternoon, giving them all our organisms for the day, It was their job then to see how they wanted to intervene and help our clinicians with the right choices. So if it was Staph aureus, because remember, we're not getting susceptibility data at this point, they would recommend vancomycin. This is just an example. For Acinetobacter baumannii, which happens to be a multi-drug resistant organisms in our institution, they would recommend starting double coverage with minocycline and colistin. And if it's not Pseudomonas aeruginosa, they would discontinue second antibiotic. So this is just a very simplistic way in which just to show you that just the organism identification alone can be pretty relevant in this setting. And I can't emphasize the fact that this is a collaborative effort. No one single person can do this by themselves. You really have to work together as a team. Now, this very nice paper came out from the Methodist Hospital in Dr. Muss's group, where they implemented the MALDI-TOF in their lab for clinical microbiology, and then they also did susceptibility testing with the BD system. And then what they did was that they had pharmacist, that is Dr. Perez here as the first author, and the lab would call her with blood cultures that were positive. And then it was really up to her to actually go ahead and contact the physician and make sure that the patient was on appropriate antibiotics and so on. She did not mind being woken up in the middle of the night. This is kind of amazing because our antibiotic stewardship team is pretty much, we give them the answers from 9 to 5, okay? And they intervene by up to 5 o'clock. We are still sending them the results, but then they intervene the following morning, of anything that's after 5 o'clock. But Dr. Perez, at least for the purpose of the manuscript, or maybe she's doing it now, I don't know, they were actually 24/7, so they were getting these answers in real time and then intervening where she would be talking with the clinicians. And you can see the result of the direct identification by MALDI-TOF on blood cultures. And you're looking at the pre-intervention period and looking at the intervention post-MALDI-TOF. You can see that the length of stay in the patients that were intervened upon were much shorter, and that there was more survival occurring in patients that were intervened on, and the hospital costs were dropped dramatically because as a result of improved time to optimal therapy, decreased hospital length of stay, and total costs with the stewardship intervention. So it really does make the point that working collaboratively, if you have a rapid assay, rapid method of detecting organisms, collaboration goes a long way. This is another paper by Vlek et al. That also looked at the mean identification time by MALDI-TOF, which is, you can see, much shorter, 16 hours as opposed to 45 hours. So, a good time difference there, more than 24 hours. And also the median time to switch to the antibiotic was much shorter with MALDI-TOF. And having the patient on an appropriate antibiotic was higher in a patient that had received quicker results with MALDI-TOF implementation. And a patient with inappropriate therapy was much lower when MALDI-TOF was implemented. So before you walk away thinking, oh my goodness, MALDI-TOF in micro is just everything and they really don't need anything more, well, that's not the case. I'm excited only because we've just not had this ability to do so much earlier. But we also recognize areas that we really need to work on. One is obviously the organisms that still cannot be differentiated, that are taxonomically very closely related. Like I mentioned, your E. coli, Shigella, we have strep pneumoniae, strep mitis. We have other gram-negatives listed here that are very relevant for us to identify and speciate, so we really do need to bear that in mind and have other procedures in place. We know of misidentifications, so if it's a mixed culture, the mixed culture will not give you a good identification. And that happens, the reason we've not gone directly live with our blood cultures from the broth is because about 7% of our blood cultures have polymicrobial organisms, and it's very hard to know if you have a polymicrobial culture, especially if you get a MALDI result. It may not be accurate, other organisms may not be identified, and if you have a dirty target plate. And then your databases is limited to as good as your databases is, so you really want to make sure you have excellent databases on hand or create your own. The instrument costs are substantial, as I mentioned. And then one of the areas that we feel right now that we are lacking is the area of middleware and software. So for example, we have a MALDI-TOF that is rapidly giving out these answers for identification. And then we have a different system, which is a microscan system that does susceptibility determination to different antibiotics. The two instruments do not talk to each other. So, which is very frustrating for us because we have to very much have it done all manually, where we have to manually, input all our answers for the ID, and then, have it merged with susceptibilities and so on. And we use the word preliminary till, that actual crossover occurs and so on. So now that there is FDA approval with the Bruker, with some of the organisms, I think Siemens is now motivated and wish to try to help us get that software, the middleware going. And the other thing to keep in mind is, potential testing downtime. So if your MALDI does go down for whatever reason, if somebody just forgot to close the hatch or didn't, appropriately, turn it off correctly, well, there's a big problem because the next person who's coming in to do MALDI is not a very happy person at that time, and that other person is not very popular. Because they did not shut the MALDI down correctly, or if the MALDI just goes down on its own, what are you going to do? Because there's so much riding on that one MALDI now that you really do need to make sure that you have really good service support. And in our situation, we ended up buying a second MALDI because we just feel there's so much riding on our first MALDI that we cannot imagine not having something to rely on. And then your susceptibility testing is not yet robust on your system. So with regards to susceptibility testing, we know we have a number of multi-drug resistant organisms that have to be dealt with in an appropriate timeline. And MALDI, unfortunately, is out there. That people are starting to work on it, especially, and there are different ways of looking at MALDI with susceptibility testing. For example, you can look at drug degradation. So if you have a drug that gets hydrolyzed because of enzymes, carbapenems or beta-lactamases made by the organism, you can look at the drug degradation. Or you can look at the protein modification as a result of drug resistance. So detection of methylated ribosomal RNA-encoded resistance for aminoglycosides can be looked at. If you have a Staph aureus versus an MRSA, can you identify specific protein peaks? That will help you identify, the organism better. Or for vancomycin resistance, Enterococcus faecium, can you look for a VAN-A-specific profile? So all of these are areas that are being looked at, along with some additional areas of research where you're looking at nutrient utilization, analysis by bacteria exposed to antimicrobials, or you're looking at quantification of antibiotics related to influx and efflux. So this is an area right now that is something that is of really high importance, that we match up our organism identification with ways in which we can rapidly give susceptibility results. And this is work in progress. So the system here just shows you have a bacterial suspension that has been incubated with your drug, and you centrifuge it, you take a supernatant, put it on your MALDI plate, and you go ahead and generate a spectra. And what you're hoping to find here is that in an organism, this is your ampicillin drug, and you have a peak here at 350, but if you have a beta-lactamase positive organism that is going to be hydrolyzing ampicillin, you have a disappearance of the peak here, and then as a result of hydrolysis, you have an 18 kilodalton shift here and a minus 26 dalton shift here as a result of decarboxylation. So looking at these specific patterns in which to identify, your beta-lactamase or your carbapenemase detection of these organisms. So in summary, we are very excited that MALDI has entered in the clinical diagnostics for ID purposes. It really has a huge potential and is already being put in place right now in terms of how it's helping us get quicker answers and for patient care. I think I've shown you some examples where you've seen improved patient outcomes and decreased hospital costs just by the implementation. The future directions, I would say, would be solid databases. We really need to rely on our databases more and have these be really relevant. And then looking at resistance and virulence markers is something that's being worked on. And then directly from clinical specimens, that's an area which right now needs to be worked on. So with that, I will acknowledge my clinical micro team at OSU Medical Center and Bruker for their support in providing us the instrument and leaving it with us for 6 months. And I'll take any questions. Thank you.
So we have time for 1 or 2 questions.
Sorry.
So, traditionally, the idea of bacteria is being hindered by when you change the culture conditions, your spectral profiles change. How is it that it's working basically? Why is that now that's solved wrong?
You mean why is the media not really playing much of a role? We've tested several different media, and we don't see the effect of, and I see where you're coming from because that was the big concern. Even today, when we try to do identification, say for mycobacteria, we use 3 or 4 different culture media, and we're constantly worried about the culture media effect. It just seems that the organism, the ribosomal proteins are just so much far more abundant that, and you're looking at a ribosomal protein aspects, you're not really looking at any other miscellaneous, and these don't seem to interfere with your pattern of organism. So it's really been something that's really been a salvaging factor, because we have tremendous number of different types of media, both liquid and solid. And if we had to do MALDI-TOF verification on each and every one of them separately, that would be a nightmare for us.
So what keeps us from doing a blood specimen or urine today? Instead of culturing it on a plate, taking a colony off, what keeps
Right.
us away from doing it?
Well, actually, some labs have already implemented that. Our lab has not. And the reason we have not, we've done about 500 some positive blood cultures for validation. So we really, bent over backwards to try to get this going, because as you can imagine, sepsis is important. Every R matters when somebody has a septic episode, directly related to mortality. So when we did our evaluation, we had about 7% of polymicrobial cultures, which means you have, instead of having one bacteria grow out, you had several different bacteria grow, which is not uncommon in bacteremia cases. When we looked at the MALDI data on those cases, it could not distinguish which one was mono or which one was polymicrobial. And I can tell you that when a report goes out to a clinician, the fear factor here is that they're going to look at that first report and say, oh, okay, this patient has an E. coli, I'm done, let's treat with this. They're not going to probably go back and look at this report on day 2, which says, no, wait a minute, MALDI just called it one organism, but there are another 3 different organisms that grew, another 2 organisms that grew. So the ability of MALDI right now to differentiate the spectra of polymicrobial organisms is not great. Okay, so that's what's kept us, but that hasn't kept some other labs from implementing it, I guess. If you can pick up the phone and talk to your clinicians and, make them aware, I think there's certainly a workaround that. Because even with your nucleic acid tests, when you have these multiplex reactions for gram-positive and gram-negative organisms, there are times it misses certain organisms. So this is not something unique to MALDI that it's not detecting. Yes, we would like better resolution of spectra, which is something that I think our companies are very aware of, that they have to build better softwares for us in which to be able to at least make a statement that this is polymicrobic, and not just walk away when you get the one predominant organism light up and the rest of them not, surface at all. So there are improvements that are definitely needed in this direction. And same thing with the urine. There is just a lot of interfering substances in urines. And that just, if you get an 85% sensitivity specificity, is that a good enough clinical-based test? We just have to make sure we are, trying to do better than what it is at the moment. And I think that's really where research has to be done directly on specimens as the next phase.
Great. Thank you very much.
Thank you.